So most of you have probably heard of a buffer at some point or another, and in this video we're going to talk a little bit about what a buffer specifically is chemically. So a buffer is a solution whose pH changes very little when acid or base is added. And that should be kind of surprising because, right, usually if you think of a solution and you add acid or you think about base, you should change the pH quite a bit by adding acid or base. But the idea of a buffer is it's designed so that even whenever you add acid or a base, that acid or a base is kind of, it reacts with components of the buffer to prevent changes in pH. So the way that a buffer is made is by mixing a weak acid and the conjugate base. And the idea is that whenever you have a weak acid and a conjugate base present, you're going to have components that can react with either added acid or added base. So let me make an example of a weak acid. So the common one is CH3COOH. That's acetic acid, right? And acetic acid has that proton, that H right there, that's going to be important in being a weak acid, right? That's the proton that is donated, is what makes it an acid. And then whenever it says the salt of its conjugate base, what that means is you would add, for instance, sodium acetate, which would be CH3COO. And honestly, with this, you don't even have to worry about the sodium, so let's just ignore it and think of it as the anion form, right? Because if you have a salt, you're going to have a cation and an anion. So you're going to have the Na, and then you're going to have CH3COO minus. So weak acid there, conjugate base over here. All right. So now, if you were to add base and you add OH minus to a solution that contains both of these, what's going to happen is the OH minus is going to react with the weak acid. So instead of OH accumulating, which would make the solution more basic, instead the OH reacts with that proton there to make water. You're going to have H plus OH minus gives water. All right. So in that sense, the buffer serves to react with the OH minus to make it so it doesn't change the pH that much. Similarly, if you add acid, and I told you in the previous video, whenever you think of acid, think of H plus or H3O plus. So H3O plus is added. That's going to react here, and H is going to be donated from here. So H3O plus you could think of as H plus and H2O. And then this H plus is going to come and react with this to form CH3COOH, and you're actually going to make this one. And then similarly, whenever this OH minus reacts with that H plus to form water, what's left is CH3COO minus. So you kind of have these two species that kind of work hand in hand to resist changes in pH. So in general, if we were looking at it from an equilibrium standpoint, so here's our reaction with acetic acid present in water, and it can break down into form, right, H3O plus and the conjugate base, which is CH3COO minus. So if you were to add more acid, what would happen? Well, the Chatelier's principle says if we add more acid, we're going to drive the reaction over to the left. And what's going to happen is we're going to make more of this weak acid. So H3O plus is going to react with the conjugate base, and you're going to form more of the weak acid. So the idea, though, is that that extra H3O plus that we add doesn't change the pH because it gets reacted. And similarly, if we add a base, right, we add a base has OH minus. So if we add OH minus, it's going to react with the weak acid, and it's going to drive this reaction over to the right. So again, the OH minus won't make the pH go up and make it more basic because it's getting reacted with that weak acid. And what's going to happen is you're going to form kind of more water molecules, and you're going to form more of the conjugate base, because once this H leaves the acetic acid, you're going to form the conjugate base. All right, so next what I want to do is kind of show you a video of this exact same thing we're talking about with the same component, just so you can kind of visualize what we're seeing, because sometimes I think this video helps quite a bit. Okay, so here is a video of a buffer. So I'm going to pause it here, and what you can see is we have a solution that is filled with both these acetic acid, which are the purple ones, and the gray on them, so you see this gray piece right here, is going to be the proton, and then the green, or sorry, the purple part here is going to be the CH3COO. So it's CH3COO in green, and then the H makes it acetic acid. And then, so that's the weak acid. And the conjugate base is going to be the acetate, and that's CH3COO minus, and that's in green. So really, the purple and the green are the same thing, which I know is weird because of the color, but the purple will always have the proton attached to it in gray, whereas the greens never will. So in a buffer, you have equal amounts of these, usually, or very close to equal amounts of these kind of circulating around. And then what we're going to do is start the video again, and we're going to see what happens whenever something gets added to the system. So you can see them moving around and with equal amounts. And then what happens is we add HCl. So we add HCl, and if you look at the graph over here, you can see that the levels of our acid and the conjugate base are consistent so far. And we've added HCl. We're not even going to worry about the Cl minus because that doesn't contribute to pH, but the H plus would. So what happens with that H plus? Well, as you can see in this kind of highlighted circle here, what happened there is we made a new purple one. And the way that we make a new purple one is that a green one reacts with the hydrogen ion, so you may basically make CH3COO plus H makes CH3COOH. So what you'll see whenever I play this again is some of the greens, which are the acetate conjugate bases, reacting with the hydrogen ions to form more of the weak acid. All right, so here we go. There's a thing, green turns to purple. And notice the main result here is that the levels of the acetic acid, the weak acid, go up, and the levels of the conjugate base go down. And it soaks up all of that H plus, and this is why it resists changes in pH, right? Because this H plus is what really determines what the pH is. Remember, the pH is negative log of the H3O plus concentration, which H plus is really the same thing as H3O plus whenever we're looking in water. So this is why the pH doesn't change, because these guys here handle the excess. All right, so we're going to let this go here, and then in just a minute, it's going to kind of reset. And in the next case, the exact same thing is going to happen, except we're going to add sodium hydroxide. We're going to add NaOH instead. So what do you think is going to happen whenever we add sodium hydroxide? Here comes our OH minus, and we can see what happens there. So OH minus is going to be a red and a gray together. And the OH minus is going to react with the acid. And basically the gray is going to come off and it's going to make water. So you're going to have gray, which is HOH, that's H2O. So you're going to make water. So you're going to make more water molecules. And in the process, the amount of acetic acid is going to go down, because as soon as it loses that proton, the purple is going to become a green one, and we're going to have more acetate. So as we play this, you'll see that the sodium hydroxide reacts with an acetic acid, converts it to water and acetate. And then exactly the opposite of what happened in the previous situation occurs. In this case, the acetate levels increase and the acetic acid levels go down. In this case, we also form water, which you can kind of see in here as well. Okay, so in the human body, buffers are very important. On a previous slide earlier in the chapter, I talked about how the normal blood pH is around 7.4. You can see here it ranges between 7.35 and 7.45. And actually, those numbers can fluctuate a little bit more depending on specific conditions. And we'll talk about that here in just a second. The principal component of the buffer in the blood is carbonic acid and bicarbonate buffering system, which again is why we talked about this earlier in this chapter. So keeping in mind that when CO2 is found and reacts with water, you're going to form this carbonic acid, which immediately spontaneously dissociates into CO2 and H2O. Or it can also dissociate into a free proton, where a proton can be donated and this bicarbonate ion. So in the end, you kind of have this kind of combination of CO2, H2O, kind of H3O plus, and then the bicarbonate ion kind of all working together. And the idea here is that in your cells, right, in your tissues, carbon dioxide is constantly being made. So constantly being made throughout your metabolism. So you can imagine the more CO2 you have, you're going to drive this whole process to the right. Think of Le Chatelier's principle, right? The more CO2, the more this reaction is going to go to the right, and eventually you're going to end up with increased amounts of H3O plus and HCO3 minus. Well, higher levels of H3O plus, right? H3O plus you think of as acid. So increased CO2 is going to lead to be more acid. So the more CO2 you have, the more acidic your blood or your solution will be, and the lower the pH will be. So what does that mean physiologically for us? So there's a condition called respiratory acidosis. And this is when your body doesn't eliminate enough CO2. So basically your body kind of stockpiles the CO2 instead of breathing it out, like is what you should do. So a lower respiratory rate, you're not breathing as much, you're not exhaling enough CO2. That's going to increase your CO2 levels. What happens when you have increased CO2 levels? You drive this to the right, and you're going to end up with a higher amount of acid, H3O plus, and a lower pH. So this could cause your pH to drop lower than that 7.4 range, and that wouldn't be a good thing. So that needs to be fixed. The opposite is respiratory alkalosis. Respiratory alkalosis is what happens when you hyperventilate, right? So when you hyperventilate, you start breathing really, really fast. So you get a higher respiratory rate. Whenever you breathe really, really fast, you're getting rid of all of your CO2. So since you're getting rid of all of your CO2, this equilibrium gets disturbed. You take CO2 out of this equation, and the process is going to be driven to the left. Well, that means you're going to be losing the amount of H3O plus, which means you're going to become more basic. When it becomes more basic, you're going to have a higher pH. So in this case, your pH of your blood could go higher than that 7.4 level. With the acidosis, it would be lower than the 7.4 level. And this is why whenever someone's hyperventilating, oftentimes they'll breathe into like a paper bag or something like that. And the reason for that is if you breathe into a paper bag, think about what would happen. You breathe out the air and then you breathe it right back in. By breathing the same air back in, you're breathing in that CO2. So this causes you not to have this large change in pH due to breathing out all of your CO2. So again, this is a real life example of what can happen due to the buffers that are in your blood and kind of processes that can disrupt the buffering capabilities in your blood.